So I'm Tim Abrahams, and this is Stefan, and so we've been asked to give you a reproductive physiology lecture. Just a thing to know about reproductive physiology, it's not that big content on your exams and things, so I wouldn't stress too much about it, and a bit of it is quite complicated, but I think you started heme recently. You should, that's the big physiology topics that you should know. So this lecture today, we want you to ask as many questions as you like. Raise your hand at any time and interrupt us. Even if you just want Stefan's number over here, feel free.
We're giving out some of these if you guys ask questions. Not if you get the questions that we ask you right, but if you ask us questions, we'll be giving out these.
And this is Stefan Dodic for all of you who have never met him. He's got a very big head. So what we're covering today, we're going through, I know you covered it all very fast and there are lots of topics, but again, they're not very high yield, all of them, so we'll go through them. And we're starting with male reproductive physiology, and that's Stefan.
So I just want to say thanks everyone for coming. It's lovely to see so many people wanting to listen to two idiots talk about reprophys. So we're really humbled that you've come here to have a listen. So just starting with the male reproductive system, it's pretty good to start with sort of the hormonal axis, as with anything, just to get our heads around the sort of the main process behind the male reproductive system. So I've got a diagram here basically summarizing it, and this is pretty important to sort of get your head around and memorize. If you're going to memorize anything from this slide, hello, memorize this. So we start off with GnRH, or gonadotropic releasing hormone, which is released by the hypothalamus into the hypophyseal portal system. Does everyone know what I'm talking about when I say the hypophyseal portal system? Tanner? Michael Tanner? Okay, so GnRH is released into the hypophyseal portal system, and it acts on the anterior pituitary gland, which stimulates the release of luteinizing hormone and follicle-stimulating hormone. Anyway. Okay, so the pathways of LH and FSH are slightly different. So we'll first focus on the LH pathway. So LH is released into the systemic circulation, and it acts on the Leydig cells, which are found in the interstitial part of the testicles. Would you like I just want something to point to the diagram so you guys know what I'm talking about. So the interstitium of the testis, so this is a cross-section of the testicle, and it works, so LH works on the interstitium, on the Leydig cells, which are found here. So Leydig cells release testosterone, and this then negatively provides the negative feedback to mainly inhibit luteinizing hormone and gonadotropic releasing hormone. There is some negative inhibition on FSH, but mainly testosterone negatively inhibits the release of LH and GnRH. I've got a slide later on just talking about the effects of testosterone and that, but suffice to say at this point in time, testosterone stimulates spermatogenesis and has various effects on the body. So focusing on the FSH pathway now, so FSH is released from the anterior pituitary. It then acts on Sertoli cells, which are found in the germinal epithelium. So this is the little amorphous looking structures here. So it acts on the Sertoli cells, and it stimulates the release of inhibin B. Inhibin B negatively feedbacks and inhibits the release of FSH, mainly FSH. There is some inhibition of GnRH, but to simplify things, it mainly inhibits FSH. So just get your head around this diagram here, and that should be good enough to get your head around the sort of the hormonal regulation of the male reproductive system. And a bit on the functions of the Sertoli cells and the Leydig cells. So the Sertoli cells, as I mentioned, they're found on in the germinal epithelium, and they basically help to stimulate spermatogenesis. So without Sertoli cells, there is no spermatogenesis. They also form an important thing called the blood testis barrier. Also, by the way, all my slides, all the stuff that's in bold, underlined, in red, in blue, in all colours of the rainbow, those are pretty important to know. And if you're going to take anything from this lecture, it's the stuff that's in underlined in bold and all that sort of stuff. So the Sertoli cells form the blood testis barrier. So this is important to know. And the blood testis barrier, these are just tight junctions between different Sertoli cells, and they're important in preventing infertility. The way this works is, you don't really need to know about it, but I just have it here for interest's sake. So it prevents the autoimmune attack on the antigenic germinal cells. So just briefly, so basically in your germinal epithelium, technically you're making a new person. So you've got half of a new person forming in there, which means your genetic profile of the cells are different, which means your receptors and your proteins are also different as well. And these proteins are technically foreign to the body, which is why this barrier is important to prevent an autoimmune attack on these cells. That's right. We forgive you, don't worry. How rude. So just a bit on the Leydig cells now. Actually, I'm gonna ask this question to our good friend Michael Tanner here. You got no excuse here. You should know this. Otherwise, God help you, mate. What do Leydig cells do?
They make testosterone and mainly LH stimulation.
Lovely. So that's basically all you need to know about Leydig cells. It's pretty simple. Okay, so just a quick bit on spermatogenesis. So this is a nice cross-section again of the seminiferous tubules of the testicles. So that's just the germinal epithelium here. And basically the way spermatogenesis works is it starts from the periphery of the lumen here, and as spermatogenesis continues, the differentiation of cells moves inwards. So basically it starts on the outside and finishes on the inside with the mature sperm. That's just your interstitium as well with the Leydig cells, just to get an idea of where things are. I have an image here of the meiotic pathway of spermatogenesis. I don't think you need to really focus on the specifics, but we'll just step through anyway. Mainly what you need to know is that spermatogonia, from one spermatogonium, we get 4 mature sperms. That's something to know. But just stepping through this diagram, just briefly covering first-year meiosis. So there are 2 pathways for the spermatogonia. They can either divide and form another spermatogonium to keep a constant supply of sperm production, or it follows down this pathway where it differentiates in the sperm. So the first phase of meiosis, DNA replication, division into secondary spermatocytes, then division again without DNA replication into 4 spermatids, and then into the mature sperm. A thing to note with the spermatids as well is that they're connected by these cytoplasmic bridges. So with the spermatids, half of them inherit the X chromosome and half of them inherit the Y chromosome. And there are some nutrients which are made from the X chromosome which aren't made from the Y chromosome, and these nutrients are important for the spermatids to mature into the mature sperms. So because of this, these nutrients need to be shared between the spermatids, so they're connected by these cytoplasmic bridges which allow for this. A bit on the epididymis and the ejaculate. There isn't really a terrible amount you need to know. I have a nice little image here of the testicle just to get your head around things as well. So this is just cross-section testicle into nice little segments, seminiferous tubules. They unify to the rete testis and then go to form the epididymis. This then continues on to the vas deferens, around here to the penis, and then out into the world. Okay. So the epididymis has 2 main functions that we need to know about: sperm storage and sperm maturation. I'll talk about sperm maturation a little bit later on in the lecture. But all you really need to know about sperm maturation is that it's a process that enables the sperm to be able to penetrate the ovum. And a little dot point there may or may not be relevant, but I put it in there for interest's sake, if anything. Actually, no, I think you need to know about it. The epithelium of the epididymis secretes various inhibitory factors which suppress the sperm and keep it suppressed until it's shot out. Did someone just say shot out? Let's be mature, guys. All right. So the ejacular fluid, not really much you need to know about here as well, but basically the ejaculate fluid, it consists of fluid from the seminal vesicles, the prostate, the bulbourethral glands, and the sperms itself from the testicles. But what we really need to focus on is that the majority of the spermatic fluid is released by the seminal vesicles, so about 60% here, which I've written. And just a diagram here giving you the anatomy, so the prostate, the seminal vesicles are found here, and this is where the majority of the ejaculate's coming from. So a bit on testosterone now, and its function. So testosterone is a steroid hormone. This means, and you would have covered it in your endocrinology learnings, but it functions on intracellular receptors, not on receptors on the membrane, so it works intracellularly. And its main functions, or apart from spermatogenesis, which I talked about earlier, an easy way to remember all the functions of testosterone is just thinking of a male and how males are different from females. But if we just roll through this list here, so hair distribution, so just think of someone really hairy, like Michael Tanner.
Thanks.
Deeper voice, so it hypertrophies the laryngeal mucosa, increased protein formation and muscle development, increased basal metabolic rate. Males have higher hematocrits than females. So testosterone has a role in increasing the number of red blood cells in the bloodstream, increased bone matrix, and minor mineralocorticoid effect. So this is actually something that all the all the steroid hormones sort of share. So your aldosterone is your main mineralocorticoid, but there are some minor mineralocorticoid effects that testosterone can carry out, as well as something like cortisol. So I've just got an image here of the metabolic pathway of testosterone. So the obvious one, so there's three of them, the obvious one is the direct pathway, so testosterone just acting on its receptors and carrying out its effects. There's also another pathway via the 5-alpha reductase. So 5-alpha reductase is an enzyme that converts testosterone to dihydrotestosterone, which then works on the receptors. So this is important, particularly in something like the prostate. So you guys have all heard of BPH? Yep. So this is actually a pathway that's been implicated in BPH, and there are some medications that act to inhibit this enzyme in some mild forms of BPH. Testosterone, so also it can be converted by aromatase to estradiol, and that works on various estrogen receptors, particularly in the brain and bone. Otherwise, testosterone is converted by the liver and excreted into the excreted via the kidneys. A few extra little bits and pieces on testosterone. So, as we know with all hormones, they are bound to proteins in the circulation. So here, the main protein that testosterone is bound to is albumin, but a large portion of it's also bound to sex hormone binding globulin. So we only have a small amount of free testosterone, and this free testosterone is a testosterone that actually performs the action of the hormone. So the negative feedback, the spermatogenesis, all the other effects that are listed here on the right-hand side. So just a bit here. Not sort of relevant, not really, but I have it in here anyway. There is something called male menopause, and I have it deliberately here in quotation marks. So we know about menopause in females and how testosterone just drops suddenly, sorry, not testosterone, estrogen, drops suddenly in the female around the age of 50. In the male, testosterone gradually declines in its levels. And this has caused sort of questioning as to whether we need to actually replace testosterone hormone in the male. But all you really need to know is the fact that it's not really recommended, and so we don't really give testosterone to old dudes.
So now I'm going to be talking about the female reproductive cycle. So just to start off with, it starts with menarche. I hope you all know that. And that's on average around 12.5 years, but of course there's a lot of variation. And what initially starts that process off is an increase in FSH and LH. And that's in puberty, and I'll talk about that a bit later. So we're going to be talking about mainly two cycles, the ovarian and endometrial cycle. And how do we get there? So there are some more hormones involved, so we're going to be talking about estrogen and progesterone. But it all starts with the HPG axis. I know you're a little bit familiar with this, but we're going to go over it again. What I wanted to focus on here, though, is I just wanted to say it sort of in basic English, because I know a lot of the time your lecturers will talk about very complex mechanisms with kisspeptin and stuff, and I don't really understand it, and I don't think you need to either. So very simply, it starts with kisspeptin. That's made in, all you need to know about kisspeptin is three small things. Firstly, where it's made, so the preoptic area and infundibular nucleus. Now, kisspeptin's role is to stimulate GnRH, and how it does that is, so there's a lot of feedback loops, but the most important thing is that it tells GnRH how fast to be released. And that's important because depending how fast GnRH is released, we either get FSH or LH. So a fast GnRH release will lead to luteinizing hormone, and a slow one to FSH. So after that, GnRH is released from the hypothalamus and stimulates the anterior pituitary. And from the anterior pituitary, we get LH and FSH. So that's just a quick overview on the hypothalamic pituitary gonadal axis. And don't overcomplicate it in your minds. I don't really think you need to know more than that. So going to the ovarian cycle, generally 28 days in length, but again, it's different for everyone. And day one is always the first day of bleeding. So that's important to have it clear in your minds. We have two phases that we're going to talk about now, the follicular and luteal phase. And that's separated by ovulation. Ovulation is what occurs at the end of follicular and the start of luteal phase. And the most important point about this is that the luteal phase is fixed. So it's always going to be 14 phase. And if you have a longer menstrual cycle, it's because of the follicular phase. So just so you can understand what's actually going on, we have primordial follicles, which a woman is born with, all of them from the start. And every month from the start of menarche, you get about 6 to 12 follicles being stimulated by FSH. And does anyone know what's actually stopping more than 6 to 12 follicles being released? Anti-Mullerian hormone. So that's basically the gatekeeper that stops more than 6. Can you give me chocolate, actually?
Chocolate. Do you want chocolate or what do you want? We have Starburst fruit juice.
Anyway, okay. So anti-Mullerian hormone is the one that stops more than 6 to 12 follicles being stimulated. And in the follicular phase, it's all about these follicles growing. And that's mainly stimulated by FSH, so follicle stimulating hormone. So as they start to grow and eventually it starts to rupture, and this is because of that LH surge, we'll talk about all of this, the ovum is released and you're left with the rest of the follicle. It turns into the corpus luteum. And the corpus luteum starts to release progesterone, and that's what dominates the luteal phase. So the next thing that I got confused with is what's actually going on in the follicle? I think this is a very neat diagram. So there's basically 3 parts to know about: the egg, the granulosa cells, and the theca. So what do all of these do? So I think the egg's pretty obvious. The granulosa cells, so FSH acts on these to convert, to make estrogen. So that's one of their main functions. The next function, it makes inhibin, and that's actually reducing FSH. And after ovulation, they make progesterone. So what do the theca cells do? Well, they make those androgens that are converted to estrogen. And the interesting part about the theca cells is that it's only stimulated by LH. Okay, so FSH actually gets pretty confusing if you actually start to read about it in depth, because it does a lot of things. So firstly, it starts by recruiting those follicles, so 6 to 12 follicles, and it helps them grow. After that, it starts to increase those granulosa cells, and that's what you might have heard as the mitogenic effect. And then because of that, because these granulosa cells are proliferating, you start to get more estrogen forming. It also stimulates inhibin, which is going to reduce FSH, and it increases LH activity. So it's a pretty confusing hormone, actually. So let's just take it slowly and go through it. The first week of the follicular phase is all about FSH. So what's actually happening here is FSH starts to rise, and this is during menses as well. So as FSH slowly climbs, we have those 6 to 12 primordial follicles stimulated, and because of that, we start to get estrogen being released, and the granulosa cells growing, and the theca cells growing. So this is when we get the, have any of you heard of the antral follicle? That's when we start to get this follicle here, and this fluid-filled sac is filled with lots of estrogen. And because of that, estrogen keeps positively feeding back, and these cells keep growing. And as the person with the chocolate now knows, the anti-Mullerian hormone is what stops more than 6 to 12 growing. Here, so estrogen keeps feeding back onto this kisspeptin circuit. What I also didn't understand at the start is, so you have 6 to 12 follicles. Well, why do we only get one follicle at the end of at ovulation? Well, basically, so anti-Mullerian hormone acts here to limit it. But after that, FSH keeps releasing more and more FSH receptors. So because even though FSH actually negatively feedback here, it actually positively feedbacks on each follicle. And what happens is one follicle starts to have more FSH receptors than all the others. And that's important because, we'll get to it in a second, but GnRH starts to be released faster. When GnRH starts to be released faster, you get a rise in LH, if you remember. Fast GnRH means more LH. Because of that, FSH starts to fall. So that's what's happening. That's what's happening around here.
Remember this graph for the exam. Remember it. It's pretty important. That's all right.
Remember the graph. So as FSH starts to fall, the follicles with the least FSH receptors start to die because they can't be sustained anymore. They need FSH to keep them growing and for that ovulation to occur so they can burst. So as FSH starts to fall, you start to get all of these other follicles dropping off until one follicle is left, which keeps releasing more estrogen. And you get this cycle where you get more estrogen being released, more GnRH released, and that means that you get this quick spike in LH. And that's how we get ovulation. An interesting thing to note, 400 follicles only ever mature to a dominant follicle. So in week 2, this is just a recap of what I said. This is leading up to ovulation. Because estrogen is now in higher concentrations, you get that positive feedback. GnRH is pulsated faster, and LH is secreted. And that causes the ovulation. So after that, you get the corpus luteum being left behind, and that starts to release progesterone. And that's what the luteal phase is all about. During this time as well, the endometrium is proliferating. And the proliferation of the endometrium is mainly due to estrogen. So about ovulation, just so you understand what's happening, so ovulation is when the follicle ruptures and you get the ovum being released into the fallopian tubes. It's caused by the LH surge, so that's an important thing that was emphasized to us in our revision as well. And it lasts for 36 hours. So after that time, estrogen starts to fall. And I think it's it's been mentioned before, but that can actually lead to migraines in women. And again, we have the corpus luteum formed, and that's the yellow body. Okay, so a bit about the luteal phase. So again, this is fixed at 14 days. That's something that you should remember. What does progesterone do? A guy feasting on McDonald's. Do you have any idea? No? It's literally written on the slides. So, Okay.
Where's some of his chicken?
So, progesterone starts the secretory phase of the endometrium. It's called the hormone of pregnancy because it's what's required. It increases vascularity and it increases the mucus secretions of the uterine secretions, and it also increases body temperature. It has some other effects that we're going to talk about in placentation. But basically, the most important thing is that it's secreted from the granulosa cells of the corpus luteum. So here, the important part in the luteal phase is that this high progesterone level and the estrogen level start to negatively feed back on the pituitary. What that means is that you get a decrease of LH because these GnRH pulses are starting to slow down. And because of that, this drop in progesterone is what causes the corpus luteum to die. So if fertilization doesn't occur, you get this decrease in LH results in a decreased progesterone, and it becomes scar tissue. And that's what's called the corpus albicans. And that's just absorbed by the ovaries again. At the same time, the endometrium can't be supported without progesterone because, as I said before, progesterone supports the endometrium. As you get that drop, the endometrium as well starts to shed, and that's why menses occurs. So the two parts of the endometrium to remember are the basal layer and the functional layer. The basal layer doesn't change during menses and stays intact, and it sits on the myometrium. And the functional layer is what changes during menstruation. Again, the proliferative phase is proliferated by estrogen, and progesterone restricts its mitotic ability. So it increases its vascularity. It's a bit confusing because it increases its vascularity, it increases secretions, but at the same time, it restricts its ability to grow. I hope that sort of makes sense. And menstruation. So a woman would lose 30 mils of blood only, and the rest of the endometrium is being shed. That's on average again. It can last anywhere between 3 to 8 days is considered normal, even 1 day. And again, it's that decline in progesterone. So, I was just going to quickly review with you. This, in the first week of the follicular phase, I just wanted to emphasize again so it's the FSH increase that causes this estrogen to increase. It positively feeds back, and eventually you start to get this drop in FSH and a rise in LH until ovulation. After that, progesterone and estrogen start to rise from the corpus luteum. This rise in progesterone and estrogen negatively feeds back, and you start to get a drop in GnRH, so the pulse is slower. This FSH starts to increase, and you get a decline in progesterone. And that leads to fertilization.
Does anyone have any questions up until this point? Is that all clear? Theo? I just got a Snapchat from you like 5 minutes ago. I hope you're paying attention to this lecture, mate.
I mean, in the male side.
Oh, and the male side. You're the male.
To be honest with you, our knowledge does not extend any further than what's on these slides. So, short answer, we don't know. But I've never seen it in the context of male reproductive physiology.
Our female lecturer seemed to think being missed out in the male part. That's why I'm asking.
Wait, so she meant
She was like, oh, why didn't they mention this in the male one? I was just wondering.
your lecturer? Oh, yeah. If she doesn't know, then we probably don't know. Sorry. Okay, so fertilization. So actually, I'm not sure. Have you guys had this lecture on fertilization? I don't know how they broke it down for you, but this is the way it was broken down for us when we were given the lecture, and it's the same way. So focusing on the 4 stages, so there's obviously preparing for fertilisation, penetration of the zona pellucida, fusion of the oocyte and sperm membranes, and egg activation. So preparing for fertilisation, so there's a bit of a focus on the male and female because those are a little different, obviously. In the male, the 2 main processes to know about are maturation and capacitation. So maturation, as I've mentioned before, happens in the epididymis and allows the sperm to penetrate the ovum. Sperm capacitation happens in the uterus and the fallopian tubes. And this is also a very important process to help with allowing the sperm to penetrate the ovum as well. So basically, maturation and capacitation, their end goals are the same. They help the sperm to penetrate the ovum, but the underlying processes within those are a little different. It's something you don't really need to focus on, although I do have 3 short dot points here on capacitation, as per Guyton and Hall. I actually recommend that textbook, it's pretty good. Which I don't think you really need to know for your exam, but for interest, for just the purpose of interest. So, for example, as I mentioned before, with the epididymis, that secretes inhibitory factors. So the fluid in the uterus and the fallopian tubes helps to wash away these inhibitory factors and give that and sort of activate the sperm. The acrosome becomes weaker, sperm becomes more permeable to out calcium, making the flagellum more powerful. So that was just for interest. I've got an image here of the sperm. So here is the acrosome, and that'll be important a little later on in terms of the penetration of the zona pellucida. Here's the head with the nucleus. The middle piece, so that contains the mitochondria, which are important for making ATP for the flagellum to move and flap around. So that's just your basic sort of anatomy of the sperm. In the female, so in the ovaries, all the oocytes or ova or whatever they're called, the eggs, they're arrested in prophase 1. This is very important. I have 2 words here, or 3 words here, which are bolded, red, and underlined. So this should spark alarm bells. You really need to know this for your exam. So all eggs in the ovary are arrested in prophase 1. So Tim talked about the LH surge just before. So this happens about 2 days before ovulation. And this LH surge is important for preparation of fertilisation. The LH surge, it does 2 things. It stimulates meiosis, so it restarts meiosis from prophase 1 onwards. It also helps to stimulate ovulation. So those 2 things, resuming meiosis and stimulating ovulation. The way it helps to stimulate ovulation isn't really important, but again, I have it here for interest's sake. It stimulates basically the production of proteolytic enzymes to weaken the follicular wall, and also helps to make prostaglandins, which we all know vasodilate, which means there's more swelling of the follicle, and that predisposes it to rupturing and shooting out the egg. So once again, once the ovum is released into the fallopian tubes, it again stops its meiosis in metaphase 2 until it is fertilized by the sperm. That image might actually be important as well. So I talked about spermatogonia and how from one spermatogonium you get 4 sperms. From one premature egg, you just get 1 egg and 3 useless polar bodies. So penetration of the zona pellucida. So basically, the process that occurs here, well, there are 2 main processes, but we'll talk about the first one, which is the acrosome reaction. So basically, what happens in the acrosome reaction? So there are various glycoproteins within the zona pellucida, ZP1, ZP2, and ZP3. And these are involved in the acrosome reaction, which, in summary, is the release of the enzymes in this acrosome, proteolytic enzymes, which help eat away the zona pellucida. Where are we? Okay. So ZP3, so you need to know a little bit about the function of the ZP proteins. So ZP3, it binds to the sperm initially, and this induces a calcium influx into the acrosome. So I have a picture here. So calcium from here to here, and that stimulates the release of the enzymes within the acrosome into the zona pellucida to eat away the zona pellucida wall. ZP2 binds to acrosome-reacted sperm. I don't exactly know what that means. It's pretty vague. It was in our lecture slides, so I've just whacked it in there. So that's basically the acrosome reaction. So maybe or maybe not you've been asking yourselves why only one sperm penetrates the oocyte. Well, this is why I'm about to explain to you. So I've got a little diagram here outlining it a little bit. So basically, when the sperm penetrates the zona pellucida and meets with the membrane of the egg, this stimulates an influx of calcium from out here into the inside of the egg cytoplasm. What this does is it stimulates the release of these cortical granules into the outside here. These cortical granules contain enzymes which modify your ZP proteins. So we talked about before how the ZP proteins are important in the acrosome reaction to help the sperm penetrate the zona pellucida. When these enzymes are modified, obviously you can't get any further sperm binding onto the zona pellucida or to help to penetrate the egg. Okay, so just a quick summary of penetration of zona pellucida. So the two main processes to know about which help the sperm do this is the acrosome reaction and the force of the hyperactive movement of the tail of the sperm. Fusion of the oocyte and the sperm membranes. There isn't a great deal to know about this. Just know about two proteins, CD9 from the egg and the Zumo from the sperm. Without these proteins, there is no fusion of the membranes and no fertilization occurs. So egg activation, the final stage of fertilization. So this involves two processes, cortical granule exocytosis and resumption of meiosis II. So starting with cortical granule exocytosis. So this is actually what I described earlier, explaining why only one sperm penetrates the egg. So cortical granule exocytosis, I'll just briefly rush through it again now. So when the sperm meets with the membrane of the egg, this stimulates an influx of calcium from the outside of the egg, in the interstitial fluid or whatever that space is, into the inside in the egg cytoplasm. This stimulates the release or the exocytosis of these granules containing enzymes which modify your ZP proteins. The other process involved in egg activation is the resumption of meiosis II. So recalling from earlier, Tanner, in which stage of meiosis, this is the million-dollar question, in which stage of meiosis is the egg in before it is fertilized? Brilliant. Do you want a lolly? So resumption of meiosis II. So this process relies on calcium transients. What we mean by calcium transients, temporary rises in the amount of calcium within the cytoplasm of the egg. So just a silly little diagram here, more calcium being shot up at various points, helping to progress the meiosis of the egg. So calcium transients. So the thing to know about this is that calcium transients are necessary and sufficient. And what do we mean by this? Well, necessary. So that means that without these calcium transients, the resumption of meiosis II cannot occur. Calcium transients are also sufficient. And what we mean when we say it's sufficient is that if you injected, for example, an egg with calcium, this will also help to stimulate the resumption of meiosis even without sperm binding to the egg. So the way these calcium transients occur, there's a nice little image here in the bottom right which sort of outlines the process. So a protein called PLC zeta, which comes from the sperm. It acts on PIP2. PIP2 is converted to INSP3. This acts on receptors within the endoplasmic reticulum of the egg, specifically the INSP3 receptor. And this stimulates efflux of calcium from within the endoplasmic reticulum into the cytoplasm of the egg. So calcium transients. Oh, this is you.
So that's probably the most important part of Repro Phys done. There are some smaller topics that we're going to go through as well. So starting with placentation. So this is obviously after fertilization. We'll go through what's happening. So if the egg is fertilized, you get HCG released, and that's what's telling the corpus luteum to keep producing progesterone. And that's until the placenta can take over and start to take over this job. Some things mentioned in your lectures, which was stressed, that I saw. So firstly, that integrins on the trophoblast cells allow the blastocyst to attach to receptors in the endometrium. And these two other points that you should probably be aware of. I'm not sure how relevant it is. So it was emphasized that the cytotrophoblast is the inner layer of the cells, and it's closest to the embryo. I've been practicing this word all day. The syncytiotrophoblast invades and digests the endometrium. Okay, so what does the placenta do? So just think of it as the hub that keeps the fetus alive. So it has three functions to know about. Firstly, it supplies nutrients, it removes waste, and it makes hormones. And we'll go through a bit of those, the most important parts as well. So it has the same tissue as the fetus and not the mother. And that's important because actually progesterone reduces the immune reaction to it. And the placenta sits in the uterine wall. So quickly about the blood supply of the placenta. So this forms by the end of the first trimester. What's sometimes confusing to people is that the umbilical artery actually carries deoxygenated blood and the umbilical vein carries oxygenated blood. But apart from that, so the diagram's pretty self-explanatory. What's important to note about this, though, is that the fetal and maternal blood supplies never mix. So your lecturer said that this wasn't going to be important, but this was stressed to us a lot in our revision, that diffusion, how we get nutrients is through diffusion, active transport, and endocytosis, and especially how we get glucose. So that's actually through facilitated diffusion. If I was going to learn one of them, I'd learn about facilitated diffusion in glucose. Apart from that, there's not much else to know. So the hormones and the placenta. So we make three. So the first thing are steroids, so progesterone and estrogen, mainly E3. So E1 and E2 need the maternal androgens to form, but E3 is the main steroid of pregnancy, even though estradiol is much more potent. Prostaglandins and proteins, so HCG is the main one. I wouldn't stress too much about prostaglandins and proteins. So steroids. So it is important to understand the steroid pathway for the fetus. E3 is the weakest, but it's the most dominant in pregnancy, as I mentioned. And it keeps increasing. So all throughout pregnancy, your E3 is going to keep increasing, and it peaks right at birth. So the placenta itself cannot make estrogen, and that's from progesterone. And that's because the fetus lacks 17 hydroxylase. And as I said before, E1 and 2 are made from the maternal androgens. So what's interesting about E3 as well is that it's created through the fetal liver. So it's a really good indicator of fetal growth and the fetus's liver function. So that's why it's sometimes measured. So how is E3 made? This is a very simple schematic. I don't really think you need to know more about it. than what's written here. So DHEAs, which is made in the fetal adrenal glands, is converted in the fetal liver to 16-hydroxylase. And then in the placenta is what's required here to convert it to E3 or estriol. I would commit this graph to memory and just have it clear in your mind. So, but the complex other pathways, I don't think it's really necessary to understand. Progesterone and estrogen. So what are their actual functions? Well, I think you've heard a lot. You've heard about the maintaining the electrical quiescence. What that really means is that it relaxes the myometrium and decreases contractility of the uterine smooth muscle. Because if you can imagine, like, what happens during labor is that we get contractions, and that's how the baby is delivered. So progesterone is important to maintain the electrical quiescence, so you don't actually have those contractions. Apart from that, it promotes uterine growth, and it can anesthetize the fetus.
We don't know what that means.
That's...
It's not our exam if it comes up.
Yeah. These are just the main things to get out of it, basically. What does estrogen do? Well, this is as vague as as it gets, but it's needed for uterine growth and blood flow. That's pretty much it. HCG. Yeah.
Can I ask a question on the progesterone? When you went through the menstrual cycle before, you said that, I don't know if you said it actually, one of you said it, and it was, you said that it was mainly for like blood flow and everything like that. Is that what you meant by uterine growth, or were you talking about it will also grow inside? Or were you talking about like the endometrium?
No, I think this is, so, this isn't about, so, the endometrium are you talking about during like menstruation? Yeah. It's not about the endometrium here.
So which part is it?
So basically, are you talking about estrogen or progesterone?
Just a uterine growth part on progesterone. The whole uterus?
I think it's this is a good question.
Put us to the test, yeah. I think we'll get back to you on that one. Right? We'll give you an answer to that question.
Yeah.
Later.
Is it the whole? I don't know. I don't want to tell you the wrong thing. That's all.
No. We'll answer that question later on. We'll finish lecture
Yeah.
and then we'll. For asking a question. Sure.
So, HCG is secreted by the blastocyst and then by the syncytial trophoblast. Again, its main function is at the start to maintain the corpus luteum and allow progesterone secretion until the placenta can take over. And it's secreted only 2 to 3 days after implantation. So you can't use a pregnancy test the day of fertilization. And that's fetal maturation.
Okay, so fetal maturation. So before birth, the fetus obviously can't do various things. It can't eat, it can't urinate, or it can't breathe. So obviously the organs involved in these processes need to be differentiated and mature once the baby pops out of the uterus. So before birth, so I got a list here of some of the examples of the maturation process and what organs are involved and whatnot. So maturation of the lungs to breathe. So the structure's changed and its function as well. So importantly, the production of surfactant. Can somebody tell me the function of surfactant? Theo. Function of surfactant. Yeah.
Is to keep the alveoli patent.
Good.
Good.
Chocolate. Okay, so preparing the gut to feed.
Whole bag. That's great.
Okay, so So preparing the gut for feeding, developing the ability to concentrate urine, generating heat for thermoregulation, and changing the hemoglobin type. So I don't think I'd really commit this list to memory. All I'd really focus on is the fact that the baby can't eat, urinate, or breathe. Think about what sort of organs would probably be involved in that. Like, for example, eating, obviously the gut, you know, urinating, the kidneys, breathing, the lungs, the hematological system. And just remember that these are sort of the organs and systems that need to be fully differentiated before the baby's born. And then I'll help you to better, not so much memorize this list, but recognizing what's wrong and what's right in a multi-choice question. There's various tactics involved in multi-choices. It doesn't really test what you know, but anyway. Eating, urinating, and breathing, remember that. So how does this maturation process occur? So it's all to do with the cortisol surge. So we have here, and by the way, this cortisol surge, so the cortisol surge, this is important in terms of the fetus and the fetal adrenals making the cortisol. So it's the cortisol coming from the fetal adrenals, which is important in helping the fetus to maturate. So I just have a graph here of the cortisol surge. So we can see a slow, gradual increase in the amount of cortisol in the circulation until it peaks just before delivery. So this cortisol surge, cortisol coming from the fetal adrenals, not the maternal adrenals, the fetal adrenals, is important in differentiating the organs and systems required for eating, breathing, and urinating. So you might wonder to yourselves, why does this happen? Isn't there a negative feedback system in endocrinology like we've been learning the whole bloody time? Well, you're kind of correct. This negative system is overridden, though, and it's sort of made redundant in this cortisol surge process. Why that happens, nobody has any fucking clue. Just accept it for what it is. And here's just a quick image of the HPA axis. Just a quick revision on the hormonal regulation of cortisol release. I won't go through it here, but this is just for your revision if you want it. So how do we know about this cortisol surge? Has someone spoken to you about the Liggins Hypothesis? Should I waste time talking? Okay. All right. I'll waste time talking about it. So the Liggins Hypothesis. So basically, how do we know about this cortisol surge? So it all boils down to this one bloke from New Zealand. He actually went to Auckland Grammar. Did you know that?
I did know that, actually.
You did? Iz was the first person. Iz was quick to tell me that as soon as he was. Anyway, here's this guy from New Zealand, and he basically carried out these experiments on sheep, on pregnant sheep. How ironic. He just did experiments. He didn't do anything else. Okay, so he carried out these experiments on pregnant sheep. And what he did was he took out the pituitary glands of the fetal lambs and he replaced the cortisol that would have otherwise been deficient from taking out this pituitary gland. And so what he realized when he injected the fetal lambs with cortisol is that they were born before expected and that they were also able to breathe. So what he deduced from this, or what he hypothesized from this, is that the cortisol from the fetus initiated labor and promoted the synthesis of surfactant. Some of this hypothesis is true, some of it is not. In terms of cortisol stimulating labor, it may happen in sheep, but not in humans. In humans, nobody actually knows what triggers labor, and that's an important point to take away from this lecture. Nobody knows what triggers labor. There are various factors involved in birth, which I will talk about, but nobody actually knows what starts this process. But what we can take from this Liggins hypothesis is that glucocorticoids are key to fetal maturation. And these are glucocorticoids from the fetal adrenals, not the maternal adrenals. So mechanisms of birth. So as Tim briefly mentioned before, when birth happens, the uterus contracts. We know that. So there are two subsets of factors involved in this uterine contraction: uterotrophic factors and uterotonic factors. Uterotrophic factors prepare the uterus for contraction, and these involve things like your contractile proteins, obviously, gap junctions. So gap junctions involved in spreading the electrical signal so that the uterus can contract uniformly and help push the baby out. Oxytocin receptors. So I'll talk about it a little later on as well. Oxytocin is another hormone important in pregnancy, in birth. Oxytocin stimulates the contraction of uterus. Pardon me. And prostaglandin synthases. Uterotonic factors induce the contraction of the uterus. So basically, these involve prostaglandins. More importantly, PGE2 and PGF2 alpha. Both of these prostaglandins help to contract the uterus. PGE2 also relaxes the cervix. So the cervix, it's this little section of the uterus down here, and this dilates during birth. So PGE2 also dilates the cervix. So obviously, we associate prostaglandins with inflammation, and there are inflammatory pathways used to make these prostaglandins. But importantly, anti-inflammatory drugs like your NSAIDs, your steroids, and all that sort of stuff, they do not prevent birth. I've got another point here in blue. So progesterone, we talked about electrical quiescence and all that sort of thing, and it sort of rings true here. So progesterone inhibits both of these types of factors, whereas estrogen potentiates them. You might be wondering how that makes sense when there's a high level of progesterone at birth or before birth. Its effects of progesterone are altered because of the changes in receptors of the progesterone. Okay, so oxytocin. So I talked about oxytocin being another important hormone involved in labour. So a quick revision of oxytocin, you would have spoken about it a little bit in your endocrinology teachings. And oxytocin, it's a hormone produced by magnocellular neurons. So I've got an image here. So these are your magnocellular neurons. They're special neuroendocrine cells whose nuclei are found in the hypothalamus, but their axons extend all the way into the posterior pituitary. So these neurons, they're important also in ADH. You guys know what ADH is? Yep. So ADH is also made by these neurons, but we'll focus on oxytocin. So oxytocin is made here in the hypothalamus, and they're stored in secretory vesicles within the axons of these neurons in the posterior pituitary. Okay, so basically what happens, we'll split this up into pregnancy and then labour itself. So during pregnancy, oxytocin is released periodically and rhythmically from the posterior pituitary, and this helps to stimulate slow rhythmic contractions of the uterus. So that happens all throughout pregnancy, and these are referred to as Braxton-Hicks contractions. What happens towards the end of pregnancy is that these contractions slowly become stronger and stronger until they're strong enough to basically start pushing the baby out and inducing labour. So during labour, an important thing to know about is the Ferguson reflex, and oxytocin is involved in this reflex. So referring to this image here, so when the baby is pushed out, it starts to dilate the cervix. There are special mechanoreceptors within the cervix which pick up this stretch and send sensory signals to the spinal cord, up all the way to the hypothalamus. The magnocellular neurons then send impulses from the hypothalamus to the posterior pituitary and release the oxytocin from their vesicles into the circulation. So oxytocin is now at a higher level, and it stimulates the contraction of the uterus. This pushes the baby further out, further dilating the cervix, sending more sensory signals, and it keeps going like that in a positive feedback manner until the baby is flung out into the world. So, so that's the Ferguson reflex. So an important thing to take note of is that it's a positive feedback mechanism. Yep. So to summarize the mechanisms of birth, so we know that they're multifactorial. There are various things and various substances and processes involved. We talked about the uterotrophic, uterotonic factors, oxytocin. So it's multifactorial. We don't know what triggers birth. Nobody has figured that out. Prostaglandins are necessary for birth. And with the withdrawal of progesterone's action is also necessary for birth. Just quickly on lactation now. So before I talk about the actual hormonal mechanisms and the production of milk and all that, just quickly get your head around the anatomy and the histology of the breast. So just starting here, so there's an extensive ductile system within the breast. So it starts first at the level of the alveoli. So these are these little sections here of alveoli. They together form one lobule. So alveoli are within one lobule. And these merge to form lactiferous ducts, which merge further towards the nipple, and this provides an exit point for milk. So this, sorry, it's distracting me. So this ductile system, it's sort of encased within adipose tissue, as you can see in this image here of a cross-section of the breast. Just a bit on the histology as well. So there's also myoepithelial cells within the breast, and they form a network, almost like a fish net around the alveoli and the lobules, so that when they contract, they can help push the milk out of the alveoli through the ducts and to the baby. Sorry. So these are the myoepithelial cells here on the histology slide. Through here as well, here. And this is just myoepithelial cells on an electron micrograph. So that's just a bit about the anatomy of the breast. So lactation, so the roles of different hormones. So estrogen is important in the development of the ductile system of the breast, so the lactiferous ducts and all that sort of stuff. Progesterone is important for producing lobules, or for stimulating the formation of lobules and the alveoli within it, and also making them secretory in nature, giving them that capacity to secrete milk. Prolactin is important for milk formation, or galactopoiesis, and oxytocin is important in myoepithelial contraction. So oxytocin, a good way to remember its function is just it's involved in contraction, and think of everything to do with the female and what needs to contract, the uterus and the myoepithelial cells within the breast. So oxytocin is important in myoepithelial contraction to inject milk out of the nipple. An interesting note here, estrogen inhibits prolactin, and this can become important in a clinical setting. So say, for an example, you have a woman who's just given birth. She needs to be able to feed her baby milk, so she needs prolactin. But what she also wants is to have, sex without a condom. So she wants the pill. So obviously we know in the combined pill, there is estrogen. So what do? You give her a progesterone-only pill so that there's still enough so that there's low levels of estrogen in the body, which means less inhibition of prolactin, which means more production of milk so that the baby can feed off of it and grow. Just the last little bit on lactation. So the let-down reflex. This is just another little reflex. So it starts off, there's an image here at the nipple. So the baby suckles on the nipple, and just as in the cervix, there are mechanoreceptors which sense movement in the nipple. This sends an impulse up the spinal cord to the hypothalamus, which stimulates the release of both prolactin to produce more milk and oxytocin for contraction of the myoepithelial cells to push this extra milk out for the baby. That's basically it.
So another important topic is menopause. So We'll start off with just the definition of menopause. It's basically the permanent loss of ovarian follicular development. And what happens is, so there's a definition that's often used that's it's the last 12 months after the last menstrual bleed in a woman with an intact uterus. That's not really accurate because it can be affected by things like if women are on the oral contraceptive pill or if they're on the Mirena IUD. So also iatrogenic causes, for example, if a woman has had a hysterectomy. So a better definition is that permanent loss of ovarian follicular development. Another point is that the testosterone decrease is usually age-related and it's not due to menopause. Through menopause, what the main hormones affected will be estradiol and FSH, and we'll go through the new hormone levels in a minute. So in Australia, the average age is 51.5, but that's actually affected by a lot of factors, even ethnicity. And what can make it earlier? Yep.
Is that a testosterone decrease in female?
There is a testosterone decrease in females, yeah. But it's not. It's normal, and it's not due to menopause. So premature ovarian insufficiency, that's defined as menopause or cessation of ovarian function before the age of 40. The common causes to know are idiopathic, so things we don't know, iatrogenic, things that we do to the patient, and a condition that's often mentioned, Fragile X. Apart from that, this is the important slide to understand. So as a woman ages, she will get a decline in follicle numbers. So the lower number of follicles can be seen in lower AMH levels. So that's one test that women can do during menopause. The decline in follicle numbers leads to a decrease in inhibin B, and because there's no inhibition, we're not going to get there's no longer inhibition of FSH, so FSH levels rise. And because FSH levels rise, you get a decrease in estrogen. I think I've tried to make these slides pretty simple that you can follow, and I think that's really all you need to know. So the symptoms and complications. So it's important to know that it doesn't just occur like at 51.5 years of age. It can occur, it starts usually even 2 years before, and it can continue. There's no start and end date. How I would learn the symptoms, you probably won't be examined on them, but they could come up in an OSCE situation as well. Just sort of use logic to figure them out. So if a woman start with hot flushes and night sweats, well, that could lead easily to sleep disturbances. And if someone has sleep disturbances, well, they might have reduced memory and concentration. And because of that, they might be irritable and anxious. And of course, sleep disturbances could cause fatigue. And these symptoms are actually really detrimental to some people as well. The other things to note are myalgia, so muscle pain, depression, and vaginal dryness. The complications of menopause. So the first thing is all about estrogen. These low levels of estrogen, what that means is it can lead to osteoporosis, and the mechanism, you covered that last semester, I think, but just to recap, so it'll affect the osteoblasts and then affect the osteoclasts. So I've skipped a step here, but I don't think you need to memorize it. So an increase in osteoclast activity is going to lead to increased bone resorption and brittle bones. Something important to note here is that the area most sensitive to this estrogen decrease is the vertebral column, and that's why vertebral cross fractures are more common during menopause. So the other effect of menopause is on cholesterol levels. So estrogen is actually protective for the endothelium for plaque deposition, and after that, there's an increased cardiovascular disease risk, so increased ischemic heart disease and increased stroke risk. Apart from that, so there's a bit of controversy about this. So weight gain itself is an age-related process. It actually has nothing to do with menopause. What menopause does, it changes the distribution of weight from, and I think it's called the androgen body type. So you get central weight gain instead of around the hips. Other complications that are common, so painful sex and vaginal dryness, urge and stress incontinence. That's very linked to menopause. I think it was found in premenopausal women was much less. And vaginitis and discharge. So that's all you really need to know about menopause. Puberty, again, is a small topic, and we'll go through it pretty quickly. So it really starts with the activation of the hypothalamic pituitary gonadal axis. That starts, it's different in males and females, but it starts at 12 years in boys and around 10 and a half in girls. Of course, this changes for everyone. And it's really marked by this first increase in LH and FSH, and that usually starts at night. So that's when it, at the start of puberty, the increase in LH and FSH is at night. So what's your growth spurt? Well, it's growth hormone plus the gonadal steroids. And what's important to understand about the growth spurt is my next point about estrogen and its closure of the epiphyseal plates. So someone who has a delayed puberty, their epiphyseal plates in their long bones are going to be, it's going to be delayed, so they get a larger growth spurt. And that's why men are usually a bit taller, because they have a delayed growth spurt as well at 12 years. And the physical changes of puberty, this is another point emphasized, but I'm pretty sure you can all figure it out for yourselves. So pubic hair, and in women, increased hip size and fat, men, broad shoulders, and a muscular physique. Last thing about puberty. Second last thing. So menarche in girls is around 12 and a half. Again, a lot of variation. And this concept of a critical mass. So it was found that women need to have a certain mass for menarche to commence. And it was shown, so with girls with anorexia nervosa before puberty, they didn't undergo menarche, and girls who had anorexia nervosa after puberty, well, they became amenorrheic. So that's all you really need to know about critical mass. So what can go wrong with puberty? Well, you can have problems with somewhere along that axis. So anterior pituitary, the hypothalamus, or the pineal gland. And what can it do? Well, it can do both. So early or precocious puberty or delayed. And one condition that's mentioned is the McCune-Albright syndrome. Honestly, I wouldn't worry too much about it. If you were going to, just know it causes precocious puberty, and it would be it would usually be a buzzword for it would be cafe au lait spots. So we now have the Tanner stages. I'd like to invite Michael Tanner to come up. Well, I think this is the most important slide of tonight, because it's a vicious cycle, starting with Michael Tanner as a young boy, pre-puberty, then the awkward phase, and finally to the man we all know and love, Michael Tanner now. I'm not going to just... I think you can just read these for yourselves, to be honest. I wouldn't commit these to memory, and I wouldn't worry too much about the Tanner stages.
Sorry.
I hope you did learn a bit more about Michael Tanner, though. And these are the diagrams that correspond to them. So this is, I think, really the most important part of tonight. If you could all go to... Have you any of you used Socrative before? Could you all go, just Google Socrative and click on student login. And we'll go through some questions. And it'll tell us.
Are we doing question 1 by 1, or are we doing 15 and then going through the answers?
We can go through that.
1 by 1. Oh, here you go. Oh. Wait, what's 1 out of 9? It means 1 out of 9.
Have actually. No, have done, actually actually answered it. These are numbers.
Yeah.
You don't need to like actually. You don't like. You could put your real name. We're not going to look at your actual dot. We don't really care.
It's all anonymous, don't worry.
Yeah.
Ryan, do you know how you a lot of people here today.
It's just no, but like so we can know when to stop. When big enough them answer. Checking their board at 70. Has everyone managed to log on? Do you need more time? You 6 people haven't answered it. Doesn't really matter. It's okay. We'll wait.
Let's just go.
Okay. How do we do? Oh, I think this tells us the answer and stuff. Cool. Yeah. So in this situation, so just going through the first question. So 55-year-old woman, so she's most likely to be post or perimenopausally, a vertebral cross fracture, and she was found to have osteoporosis. So clearly you all understood my chart. During menopause, because of the lack of inhibin, we get an increase in FSH. Beta HCG, progesterone and estrogen. Well, estrogen's going, which hormone will be elevated? Estrogen will go down. AMH is going down as well. Beta HCG is during pregnancy. So, is that clear? Anyone have any questions? Okay. You guys can all answer this one.
All right, it seems like you've all answered the question. Okay, so pretty much the majority of you answered correctly. So just ruling out all the other options. So spermatogenesis. So this is slightly vague. Leydig cells are technically involved in spermatogenesis because they make testosterone, which is involved in spermatogenesis. But the better answer would be androgen production, as testosterone is an androgen and it's made by the Leydig cells. Spermaturation, that's in the epididymis. Capacitation, that's in the uterus and the fallopian tubes. LH production, that's the anterior pituitary. So pretty simple question.
Okay, so we'll go through this one. This one was a bit more. This one was a bit more tricky. Yep. So that's correct. So 50% of you got that. So estrogen causes the fusion of epiphyseal plates. So does everyone know why that's actually true? Or I don't know how to. Well, yeah, I'm pretty sure it just causes. Testosterone is responsible for skeletal long bone growth.
So, no, testosterone is involved in skeletal and bone growth indirectly when it's converted by aromatase into estrogen, and that acts on bone receptors to stimulate its growth.
Adequate dietary protein. Responsible.
I mean, if you eat more, you're going to grow more, so...
Yeah.
And Jay, was there any confusion? Did anyone, what was the confusing point about... For those of you who answered D...
The word false.
Sorry?
The word false? Yeah.
Oh.
That's true. That's a good point.
It's a common mistake.
Does anyone have any other concerns about this? Any other confusion? It's more, these questions are more of a process of elimination, and I think this is a bit on last semester stuff. Okay. We'll move on. What is the site of sperm maturation?
Sperms is a word. Thank you.
Yeah, that's pretty bad.
I don't think we really need to explain this question. Majority of you got it right. Yeah. All right, how did we do? Okay, that's pretty.
Okay.
All right, I don't think we need to explain that question either. It seems like the majority of you got it.
really tough. I'll throw it on last about the fact that I mentioned it.
What are we showing?
What are you worried about? I don't know if someone just listens to it and thinks fills me up for like level 1. My meme. Why world YouTube?
Okay. So, so for the people who put progesterone,
So basically, all we really need to know to answer this question correctly is that estrogen inhibits prolactin, which would inhibit milk production. So obviously, that's the answer. Next question.
Did you talk about being a therapist? What does it say? Do you remember? You explained it. Answer the question.
as friends. As friends. As friends, you have it's not increasing. It's not increasing.
Alright, stop.
Okay. So, just in case you were wondering, so normal menstruation can be between 1 to 8 days, and it can be up to 35 days before it's considered abnormal. Next question. Oh, shit. Okay.
Where'd you go? You didn't ask us any questions. Make it rain, something to do with it. Dude, I gotta sit down.
All right. Okay. So, just if you'd like a way to remember this, umbilical arteries. So the two umbilical things, are the ones that are the wrong way around. They carry deoxygenated. So arteries carrying deoxygenated. The uterine vein carries oxygenated blood from the placenta to the mother. Let me just get the picture up for you. That might be easier. Yeah, so the umbilical artery is going from the fetus to the placenta. The uterine vein is going from the placenta to the mother. And the artery from the mother to the placenta. Do you want to hand them? What do you want? We have Turkish Delight and Boost.
Oh, why don't you go through the question?
Literally just.
Okay. We'll just get to the end. Like, we'll go
Just come down after these questions and grab all the chocolates and stuff. Every man for themselves.
Exactly, that's what we want. No one's going to listen. I just had an intermission. Take it down.
Oh, you can throw space.
You done? All right. We'll go through this. So, okay, everybody. Quiet. Shh. Everybody. Okay, so all right. So basically, to get this question right, it's just a matter of memory, really. There's nothing really else involved in it. So facilitated diffusion, glucose. That's how glucose is transported. Mainly through that, you guys have heard of like the sodium glucose co-transporter. Yeah, it's basically that. So that's the process of facilitated diffusion, which is the energy made from the sodium moving through the receptor is also used to push glucose through. Passive diffusion. What goes through passive? I don't know. I can't remember. Yeah, passive diffusion, water and urea, active transport, amino acids, receptor-mediated endocytosis, stuff like cholesterol and stuff like that. So this is just a memory question. There's nothing else to it.
Why is C wrong?
Okay, guys. Answer the question. We have 5 more to go through. Give it a shot. Okay, so we'll go through this one. So, if you remember, it actually stimulates the corpus luteum to make progesterone, not estrogen. Why this one is wrong? Because it's not really the ovarian follicle anymore. And it's not detectable at the time of fertilization. It's 2 to 3 days after. So what is the dominant estrogen in pregnancy?
Still be going. I doubt it. Should check. I doubt it's going to be
Okay, so let's go through it. The dominant, so it means the most. So E2 estradiol is the most potent. And you guys, yeah. And estradiol is the least potent but the most dominant. And it increases, keeps increasing all through pregnancy until term. All clear? Any questions so far with any of the questions? No? Either these questions are too easy or we're really good lecturers. So, which of the following regarding progesterone in pregnancy is true? Okay, guys. Keep going.
Ask me if there's anything on the clarifying come down here at the end.
Yeah, yeah.
I mean, when we're finished with that, just come down and otherwise.
Okay. So we'll go through this one as well. So, progesterone actually maintains the electrical quiescence, which means that it relaxes the uterine smooth muscle. It relaxes the myometrium. Yeah, so this one is one of those dot points that I had. Has a level which is 10 times lower than estriol. Yeah, I don't know, but that's clearly wrong. A lot of this is just elimination and knowing which ones are right. So it causes relaxation of the smooth muscle exclusively in the genital tract. Well, it's not exclusively in the genital tract.
All right, the majority of you have answered the question. Just for the sake of time, we'll speed this up a little bit. We only have 2 questions after this, but yeah. Okay, so it seems like the majority of you got it right. So let's just go through this step by step, go through each of the options. So uterotrophic factors are those which prepare the uterus for contraction. That's true. So the answer is not that. So the double negative in the false question, that's sometimes confuses people. B, okay, so that's obviously true. Glucocorticoids can be, sorry, it's false, which means it's the correct answer. Glucocorticoids can be administered exogenously to induce labour. That's wrong, because no one actually knows what triggers labour in humans. So nobody knows. That's something to really take away from this lecture. If you're going to take away anything from this lecture, take away that. No, there are other things you need to know, but... in general, reproductive physiology is pretty low yield, but anyway. All right, so C, uterotonic factors. So that's correct. That induces contraction, so it's not the answer we're looking for. D, prostaglandins are uterotonic factors. That's true, so it's not the wrong answer. Progesterone inhibits both uterotonic and uterotrophic factors. Estrogen potentiates them. That's true, so it's not the right answer, so therefore we get B. We got 2 more questions, guys, and then we'll be out of here. We know, like, your mind's probably saturated by all the reproductive phys that we've been telling you about.
0, 1. Was it children? Not true, because they read it.
Ten more seconds. Okay, so the majority of you got this question right as well. It's pretty self-explanatory. I think this is a pretty easy question, to be honest. Sorry to the 3, 5, and 6% that got it wrong.
Next year as well.
The return. Just invading everyone's medicine.
Turning everyone into those rubbish practices. Alright, just go.
Okay. Okay, guys, this is the last one. And as 52% of you have realized, it's really clever because there are 2 correct answers here. Hey. Yeah, so these last 2.
Whether we intentionally did that is probably not the case.
Which one would I have put? I would have put E because that's the most obvious one. D is also, if you listen to Stefan, is true. I'm glad that none of you put that. Sorry about this question, but I hope you guys learned a little bit, enjoyed yourselves, and thank you for putting up with us for since 6 o'clock.
If anybody has any questions or anything, feel free to come down now and ask us, because we'll stay for a little bit.